Hey Mustangs, in this video we're going to be taking a look at X-linked gene disorders. So a quick introduction to see how X-linked gene disorders are going to be different from autosomal gene disorders. So in order to do these notes, there's no handout for this one, so you're actually going to write these directly into your notebook, specifically page 89. So page 89 is where you're going to go to write down these notes. So let's go ahead and get started here. So we're looking at X-linked gene disorders. Okay. X-linked gene disorders. So we've already talked about gene disorders, but we didn't talk about them in the form of X-linked. We talked about them as autosomal. So if you think about a karyotype where you have all 46 chromosomes laid out from longest to shortest, sex chromosomes last, autosomal gene disorders are when there's a mutation within the genes anywhere in the first 44 chromosomes, or 22 pairs, you could say. Here, this is specifically connected to the X chromosome. And what we learned here can apply to the Y chromosome as well, except for the fact that anything that is found in the Y chromosome would only apply to males and not to females, whereas X-linked disorders can affect both. Okay? All right, so if you picture the X chromosome, there's about 1,184 genes in the X chromosome. Okay, so X and Y chromosome, even though they have their own special name, they're not pair 1, pair 2, anything like that, they are still chromosomes. When you look at it, it's still DNA wrapped around histone protein. So the only thing that makes it special is that within the DNA, there's sections that have instructions for making proteins that turn you into a boy or a girl. But it's still simply just DNA wrapped around histone proteins. But they get the names X and Y because of their connection to the determination of sex, male or female. Okay? So an X-linked gene disorder, spelled it right that time, is when one or more genes are mutated within the X chromosome. Okay. Now, in class, we're actually going to work out Punnett squares and stuff for this, but I just want to do a brief intro to understand what's going on here. So, to really understand gene disorders, we have to understand the genotypes that are possible with these disorders. Okay, so for a female, remember, a female has 46 chromosomes total. 44 of them are autosomes. Remember, autosomes are just chromosomes that aren't the sex chromosome. So if it's not a sex chromosome, then it's automatically an autosome. Or if it's not a sex chromosome, it's automatically an autosome. Then females have an X and an X chromosome. So females have 2 X chromosomes. So that is what makes a female. Whereas males have an X chromosome and a smaller Y chromosome. And that's what makes a male. So a female would have 44 autosomes plus 2 sex chromosomes. That would equal her 46. A male has 44 autosomes plus his 2 sex chromosomes, which equal 46. Okay, so in both cases, you still have 46. And these are all the same from humans to females when you're talking about the autosomes. The instructions within those autosomes might be a little bit different, but the genes are all in the same place and all that. So when we talk about X-linked gene disorders, a female could have multiple karyotypes here. Multiple genotypes. So we're going to use big N equals normal, little n equals disorder. So this is very generic here. It could be any disorder that we're talking about. And most of the X-linked gene disorders, thankfully, are recessive. Okay, so big N would be the normal, okay working allele, means it makes a protein that works and does its job. Little n means it makes a mutated allele that doesn't do its job. So keeping that in mind, a female, when we write the genotypes, instead of just writing big N, big N, big N, little n, little n, little n, you have to write the sex chromosome itself. So you write X, big N, X, big N. That's one possibility a female can be. that means her phenotype would be normal in this case. Okay. Another possibility for a female, she can be X big N, X little n, so she's heterozygous, she's still normal. Because many disorders, as long as you're making one, you have one gene that's making working proteins, you're usually fine. So here's normal. But we're going to put a star here. This person is considered a carrier. Okay, so always remember, and we saw this with even autosomal gene disorders, if someone is normal but they're carrying the mutated allele and they can pass that mutated allele on to their children, to their offspring, then they're considered a carrier. Okay, so carriers are always heterozygous. Totally hard to spell this thing. So you have a carrier here. This is a female who is carrier. She's normal, but she carries the allele inside. And then the last possible genotype a female could have is little n, X little n. So, and remember, this is one from each parent. So this is one from mom, one from dad, one from mom, one from dad. It could be switched, one from dad, one from mom, doesn't matter. One from mom, one from dad. And in this case, this person would have the disorder. Okay. So these are all the possible genotypes for a female and what she could pass on. And then here on the other side, we'll do genotypes of males. Genotypes of females. So the genotypes of males. Males, remember, have an X and they have a Y. So a male, he can have an X with a big N. In that case, he would be normal. He would not have a disorder. He carries the allele that makes a working protein. And when you look at X and Y, here's the thing that you have to always remember. Whenever a male gets an X, it's always from his mom. He gets the Y from his dad always. Why is that? Females can only pass on X's. Males can pass on an X or a Y. A female can never ever pass on a Y. So when you look at a male, one always has to come from mom, one always has to come from dad. So when you're looking at X and Y chromosome, the Y always comes from dad, the X always comes from mom. So X big N equals normal. Then the other possibility is X little n Y, and in that case, the male would have the disorder. Okay. For males, this is the only two possibilities. Now, because of this, males are more likely to show X-linked disorders because males only have basically a 50-50 chance. They either have the disorder or they don't. Females, we get 2 chances of not having the disorder and only 1. So 2 out of 3, we're not going to have the disorder, and only a third have the disorder. So males are more likely to have X-linked gene disorders than females are. Now in class, we're going to work out some Punnett squares with this and see how these X-linked gene disorders will be passed along and the chances of passing them along.